Humans, like all species, have shared physical traits. We also have unique combinations of traits, such as eye color, height, and gender. These traits and their variations help make every person distinct. The passing of traits from parent to offspring is the process of heredity. Scientists began studying hereditary patterns in the mid-19th century, and in the early 20th century, they developed tools to study genes, the chemical factors that determine traits. Heredity can be studied at the cellular level. Multicellular organisms like plants and animals can be made up of billions of cells. Hereditary information is contained in the nucleus of each cell as molecules of deoxyribonucleic acid, or DNA. A DNA molecule has two long strands of sugar and phosphates that form a double helix, or spiral structure. These strands act as a kind of backbone for the molecule. They are bound together by pairs of bases that resemble the rungs of a ladder. A sugar, a phosphate, and a base together make up a molecule called a nucleotide. A strand of DNA can be thought of as a series of stacked nucleotides. Nucleotides make up the genes that contain a person's hereditary information. A nucleotide can have one of four possible bases: adenine, A, cytosine, C, guanine, G, or thymine, T. These bases occur only in specific combinations. A goes only with T, and C goes only with G. Hundreds or even thousands of these bases in a long chain make up one gene. Genes contain instructions for assembling the proteins in a cell. These proteins control and regulate all the processes of the cell. They are capable of performing a wide variety of functions. Some proteins build cells and tissues. Some are enzymes that catalyze chemical reactions. Others help in the digestion of food or carry oxygen in the blood. Genes dictate the formation of proteins to control the functions of an organism. The four types of nucleotides may not seem like enough to make all the necessary proteins for an organism, yet the varying sequences of these four nucleotides are more than enough. DNA's chain-like molecules are very long, so they are coiled and folded to fit inside the cell. The combination of DNA and several proteins is called a chromosome. Chromosomes vary in size and shape and usually occur in matched pairs. Different species have different numbers of chromosome pairs. Goldfish have 47 pairs. Some orchids have 28 pairs. Guinea pigs have 32 pairs. And peas have 7 pairs. Humans have 23 pairs, or 46 chromosomes per cell. In many species, there is a pair of chromosomes that determine gender. In humans, females have 2 X chromosomes, while males have an X chromosome and a shorter Y chromosome that has fewer genes. The long strands of DNA are compacted into chromosomes by spool-like proteins. DNA wraps around each of these proteins, forming a series of loops. Humans have approximately 20 to 25,000 genes, referred to collectively as the human genome. Except for identical twins, every member of a species has a set of chromosomes with its own unique DNA. This is what gives us our unique set of traits. Our genes, located in chromosomes and formed by DNA, make us stand out in a crowd. Did you know? There are 2 types of twins, dizygotic and monozygotic. Dizygotic or fraternal twins occur when 2 eggs are fertilized simultaneously. Monozygotic or identical twins occur when a single fertilized egg splits at an early stage to become 2 embryos. Approximately 1 out of 80 births results in twins, not including births assisted with fertility drugs.
When an organism's cells multiply, they usually have the exact genetic information as the original cell. The same is not always true for reproductive cells. For many organisms, genes recombine with every new generation to enhance genetic variation. Every cell nucleus in a multicellular organism has nearly identical genetic information. However, cells may become specialized to accomplish specific tasks. Plants have leaf, root, and trunk cells that are structurally quite different. Human bodies have approximately 200 types of cells. How can cells develop in different ways and still have almost identical DNA? Genes make possible the sophisticated formation and regulation of cells. They contain instructions for individual cells to develop specialized functions. Shortly after conception, an embryo has many stem cells which have the ability to become different specialized cells in the organism. Stem cells in an embryo eventually become all the various types of cells, like nerves, muscles, and blood cells, and also form organs like the stomach, pancreas, and intestines. Genes control this process as the organism grows. The complicated differentiation of cells is built into the genetic code of every cell of the organism. When cells multiply, it is vital that genetic information is passed on to the new cells. Most plant and animal cells undergo mitosis, a process of cell division. Before mitosis can occur, the DNA in a cell must be duplicated. This duplication occurs during the stage of a cell's life called interphase. During interphase, the DNA double helix comes apart like a zipper. Each strand replicates itself so that the nucleotides and gene sequence are repeated exactly in the new strand. This makes 2 copies of each chromosome called sister chromatids. The cell now contains 2 complete copies of its genome. After interphase, mitosis can begin. The first stage of mitosis is called prophase. The sister chromatids condense and thicken, and the nuclear envelope, a membrane that encapsulates the nucleus, disintegrates. Now, the chromosomes are visible under a microscope. Sister chromatids line up along a central axis during metaphase, the second stage of mitosis. At the beginning of the anaphase, the third stage, the sister chromatids separate at the centromere, and each half of the pairs move towards opposite poles. The final stage is telophase, in which a nuclear membrane forms around the chromosomes at each pole. When mitosis is finished, the cell must completely divide into 2 separate cells. The process where the cell divides at its center to form 2 daughter cells is called cytokinesis. Each new cell has a matching set of chromosomes and a full set of cellular structures. This is how specialized cells multiply to form tissues and organs. Bacteria and other single-cell organisms reproduce using mitosis. In this process, the 2 new cells created are usually genetically identical. If multicellular organisms only used mitosis to reproduce, their offspring would be very similar to each other because their genetic information would usually be identical. There would be less variety among family members. Plants and animals reproduce sexually. 2 sex cells, one from the male and one from the female, fuse to make a new cell. Half of its genetic material comes from the father and half from the mother. This is called fertilization. Meiosis is a special type of cell division that occurs in the reproductive organs of both sexes. Prior to meiosis, genetic material doubles the same way that it does before mitosis. Chromosomes replicate and form X-shaped sister chromatids. Then something different happens. Each chromosome lines up with its corresponding partner, and they exchange genetic material. This is known as crossing over. The chromosomes collect in the middle of the cell, and one chromosome from each pair is pulled to an opposite end, and the cell divides in two. Both new cells contain half the number of chromatids. Each cell divides again, splitting the chromatids in half. The process creates 4 gamete cells, reproductive cells containing half the necessary DNA. If a female organism produces these cells, the gametes will be the ova, or egg. In the male, they are the sperm. Fertilization occurs when the egg fuses with the sperm. When they fuse, the new cell is called a zygote. This cell now has a full set of genes for development into an adult, since it gains half the necessary amount from the mother and half from the father. Then the single cell divides by mitosis and eventually grows into an adult. An average human starts as one cell and becomes in a trillion cells. The incredible number of gene combinations from this type of union creates a species and the uniqueness of nearly every individual. Did you know? Stem cells are present when a human embryo is developing, but they are also present in adult humans. Bone marrow contains cells called hematopoietic stem cells, which generate red and white blood cells. Stem cells are also present in the skin, brain, and small intestine.
As traits are passed down through the generations, mistakes in the copying of DNA may cause diseases. Scientists studying human genes are working to find cures for genetic disorders. People have known for centuries that they inherit physical traits from their parents. But until relatively recently, scientists had no tools to examine genes and determine patterns of inheritance. In the mid-1800s, the Austrian monk Gregor Mendel conducted a series of experiments on peas that led to the foundation of modern genetics. Mendel's scientific curiosity drove him to study the heredity of pea plants in his monastery's garden. He systematically crossbred plants that had different traits. Mendel bred a plant that produced smooth peas with one that produced wrinkled peas. He expected the next generation to have a combination of the two traits. Yet each time he crossbred them, all the offspring produced only smooth peas. Taking the experiment one step further, Mendel crossbred that group of offspring, producing a second generation of crossbred plants. The results were also surprising. Some of these plants produced only smooth peas, while other plants produced only wrinkled peas. Mendel realized that something was carrying these traits on to subsequent generations. Mendel reached the conclusion that when an organism has two different traits, one is dominant and the other recessive. With the pea plants, smooth is a dominant trait and the wrinkled is recessive. Mendel also discovered that while the dominant trait might be expressed in one generation, the recessive trait could be expressed in a later generation. Human brown eyes are dominant over blue eyes. So how can two brown-eyed parents have a blue-eyed child? The answer is that both of these parents carry the dominant gene for brown eyes and a recessive gene for blue eyes. If either passes on the dominant gene, their child will have brown eyes. If both parents pass on their recessive gene, the child will have blue eyes. We know that Mendel's inherited factors are the result of the absence or presence of genes that produce those specific traits. Each parent contributes half the genetic material to the new organism. The thousands of different genes with dominant and recessive traits combine to make unique organisms. Occasionally, in the copying and recopying of genes, errors occur in the sequence of nucleotides. These errors are called mutations, and they can cause serious diseases. Cystic fibrosis occurs when missing nucleotides prevent a gene from making a functional protein. Down syndrome is the result of an extra chromosome 21, resulting in a total of 47 rather than normal 46 chromosomes. Sickle cell anemia, a disorder of red blood cells, is brought about by a one-letter misspelling of a base pair, meaning that one nucleotide is incorrect. Cells do, however, have ways of correcting errors. A type of proofreading function scans the DNA and corrects any base pairing mismatches that it finds. Incorrect nucleotides are removed and replaced with the correct one. Some mutations occur spontaneously, but others come from exposure to physical or chemical agents. Cancer is a mutation of the mechanism that regulates cell growth. It can be caused by cigarette smoke, ultraviolet rays, exposure to asbestos, X-rays, or other natural and environmental causes. When a cancerous cell divides, it repeats the cancer in each new cell and spreads the disease. Not all mutations are destructive. Some can help a species adapt by increasing its genetic diversity. Mutations in bacteria help them become resistant to antibiotics. Understanding how genes work helps explain the complexity of cell structures and processes. In 1990, scientists began the Human Genome Project. Their goal was to identify and map the thousands of genes in human DNA. It will ultimately help biologists understand the way genes control cells. Armed with this information, scientists hope to develop medical treatments for genetic disorders. Their work may lead to the ability to predict or prevent diseases. In spring of 2000, after just 10 years of work, that they had finished mapping the human genome. They published their findings in February 2001. The teams discovered approximately 20 to 25,000 genes in the human genome. They had expected to find over 100,000. Scientists now have a detailed blueprint of our genetic code. Their next step is to research patterns and relationships among the genes. As promising as it is, genetic research raises many ethical issues. It has become possible to modify the genes of plants and animals, including humans. Genetically modified plants show resistance to disease, but some are concerned that they might overwhelm native plants. Many are worried about the ethics of tampering with nature and how human health might be affected. Parents may be able to select traits in their children. The long-term implications of these scientific findings are yet to be discovered, but most likely, coming decades will move much further into the science of genetics. Our descendants will be greatly affected by the choices we make now, and the power of genes will change our world forever.
Jack and Lisa Nash want to have a healthy baby, but they want something more. A baby with matching bone marrow selected to save their daughter's life. It's never been tried before.
We need to do it to save Molly, and I think we need to do it to make sure that we have healthy babies so another baby isn't brought into the world to have to go through what Molly has gone through. And she's no more worse for the wear, but she is. 80-year-old people haven't gone through in their life what Molly's gone through in 2 years. And she's a strong cookie, and that's good, and she's made us all stronger. Right?
This man's genetic secret is known only to his family. In his 30s, he will most likely begin to lose his motor and cognitive ability to Huntington's disease, a genetic illness. He and his wife want to have children who will be free of the Huntington's gene.
I always wanted to be pregnant and have a child. But the more I learned about the disease, the more I knew that I didn't want them to have to go through that. You know, we prayed about it a lot before we ever did it, but getting the news that they were Huntington-free, that was very exciting.
For people with genetic mutations, having children has long been a dangerous game of chance. But new genetic diagnostic techniques are offering more hope than ever before.
Using in vitro fertilization-related technology, they can now see whether the embryo and fetus will carry this genetic defect or not. So you could take the abnormal embryos out and only transfer back the normal embryos.
To make a genetic diagnosis, an acid probe cuts through an embryo's wall and a single cell is removed. The embryo usually has 8 cells at this stage. Inside the cell's nucleus are 23 pairs of human chromosomes, and inside them the 20 to 25,000 genes that make up the human genome. Scientists can identify the genes that cause diseases by using information in the Human Genome Project. Mutated genes cause a variety of diseases, such as muscular dystrophy and hemophilia. Fanconi anemia is a genetic disorder that leads to the failure of bone marrow. Jack and Lisa Nash unknowingly passed a damaged Fanconi gene to their daughter, Molly. Unless likely to develop leukemia and may die before the age of 8.
She was born on the 4th of July, and the nurses didn't know what to say. The doctors don't know what to say. You know, nobody knows what to say.
When it happens to you, it's a whole different thing. This can't be happening to me. I didn't do anything while I was pregnant. How could this be happening to me? What did I, you know, you want to ask, what did I do to deserve this?
We had a good number of ultrasounds during Lisa's pregnancy, and they didn't see any signs of this. I mean, she's missing her thumbs. I mean, you'd think that they would notice those things.
When I saw her, I apologized to her. Told her I was sorry. But I thought she was the most beautiful baby I'd ever seen. And I loved her the instant I saw her.
She was a good kid.
She looks like me.
Yeah.
You want to sit up so you don't throw up, please?
No, I don't.
I would really appreciate it if you didn't throw up.
No.
Molly has to be fed directly through a valve into her stomach. Digestive and heart problems are classic signs of Fanconi's, as are deformities of the arms and hands.
Oh, it smells lovely. Yeah. Oh, does it smell good?
Hi, Molly. How are you?
I waited and waited for a grandchild, and I knew I would fall in love with the grandchild and, of course, hope that everything would be healthy and perfect and normal. But I did not know how deeply I was going to fall in love, maybe even more so because of all the things we've had to go through with her. We all call her the baby goose. I'm not sure why or where that came from, but it's kind of a nickname. And yes, she touches everybody's life that she's around. She's constantly smiling and kissing you, and she's special. She's just extremely special.
The Nashes have searched far and wide, but no bone marrow donor has been found. Molly's best chance is for the Nashes to conceive a brother or sister with matching marrow. Lisa and Jack consider getting pregnant.
Our problem with having another child is, what are our chances of having another child that has Fanconi's anemia?
There's a 1 in 4 chance that the baby could have Fanconi's again, because this is an autosomal recessive disorder. There's about a 1 in 5 chance that the baby would be a bone marrow match for Molly. The chance of having a bone marrow match and a healthy baby together is a lot lower.
Neither parent likes the idea of having several births, given the odds for a healthy baby that was also a bone marrow match. Molly may not have enough time for that.
She said that her brother...
What can we do to beat these odds? What technology can change this? And you keep looking.
Could modern medicine help?
They called and asked whether pre-implantation testing was available. We were doing our homework. They called back again and they said, you could identify which ones don't have this lethal disease. And then if there were any that were an identical match for our daughter, you could transfer those as well, preferentially stack the deck in the favor of our daughter. And now, at the time of delivery, you could hand this couple a healthy baby without this dreaded condition that you'd normally throw away, and simply give it to the sister and cure the disease in her simultaneously.
The Nashes were asking Mark Hughes for a newborn healthy baby and more.
Get him. Can you give me a nose kiss?
A baby to save Molly's life.
A nose kiss.
Now, on the surface, that sounded like a wonderful way to use this technology, but the more I thought about it, the more I became concerned. Because, for me, it was crossing this line between testing for serious, lethal genetic disease and testing for a trait, in this case, HLA. A trait that is of no intrinsic value to that newborn baby. It's of value to somebody else, Molly. And is that the right thing to do? Is the raison d'etre of that second child to save the life of the first, and not for its own sake?
I'll use the old-fashioned remedy.
HLA, or human leukocyte antigen, stimulates the immune system.
Thank you.
In order to save Molly, the embryo will need to be an HLA match. The benefits are obvious, but the procedure raises a question regarding all genetic manipulation. Where might it lead? Dr. Sue Daniels is a senior embryologist involved in the production of IVF embryos.
Every step is bold, and before anybody has really thought about what. Some people may find that unacceptable. Others, it doesn't really matter, and I'll accept the risks of the technology. But certainly, the technology is running far ahead of what any ethical considerations, any social implications, any of that has been considered.
Eminent biologist Ruth Hubbard is also concerned.
When in vitro first was developed, there were numbers of us who were extremely concerned that it would open the door to the kinds of developments that have, in fact, taken place.
I'm sure some people think we're playing God. Because you're choosing what you want.
And what would you say to them?
I would first want to know if they were in our situation.
Yeah, I would tell them, I hope you're never in my situation, but don't judge me until you walk in my shoes and know what it's like to live through what I'm living through. And to hug your healthy children and thank dear God that they're fine. But to not criticize me or ridicule me for the choices that I have to make to save my child.
When the Denver University Center for Reproductive Medicine realized the complexity of the ethical issues, they called a halt to the procedure. A medical review board was convened. The Nashes held their breath.
We don't have any other choices. A family who wants to have a healthy baby, who wants to save their own child. What other choices are there?
Mark Hughes' work went on, despite new cutbacks in his federal funding.
So I'll be
he would not withdraw the treatment from families already in his care. Has learned that the ethics board had given permission for the treatment to go ahead.
Okay, well, this is good. At least I know something. Okay, and then I will call you on day 3 and come get my blood drawn. All right, thanks a lot. Bye-bye. Mark Hughes has still not said that he knows how to do it.
He'll figure it out, Lisa.
And he says I can drink champagne on New Year's Eve.
You don't need to drink champagne.
Now Lisa must start the IVF treatment. This has become a common technique for infertile couples over the last 20 years. It requires a heavy cocktail of hormones.
He wants to take my eggs, do the egg retrieval on the 6th of January. Prior to that, he would probably start me on the Pergonal the 27th of December. That's the stimulating drug. Then what he said was he'll put me on Lupron day 21 of my next cycle. So a month from now, he'll put me on the Lupron. I'll stay on that until they do the egg retrieval.
I'll do it.
But in addition to the Lupron, I'll be on the stimulation drugs. And I'll be on the stimulation drugs for 8 days. Then I'll get HCG on the 9th day. They do their egg retrieval 35 hours later on the 11th day.
These women are definitely at risk because of the manipulation of their hormones, because of the hyperstimulation, because of the extreme level of hyperstimulation.
Fine. She's fine.
I wonder whether these women really are aware of the enormous risks they're putting themselves at, and what are they doing for those children if it in fact turns out that they themselves are putting themselves in mortal danger.
Where are your shoes?
Two months have passed. Dr. Mark Hughes has finally found the needle in a haystack, the damaged Fanconi gene. Tomorrow, Lisa Nash's 5-day procedure begins. She has been on maximum hormone stimulation.
And it feels uncomfortable at first.
Yeah. Like if I go over a bump, you can feel your ovary kind of going like that.
Oh,
it's gross. But I can't pants because my tummy's so puffy. And I won't put on my maternity clothes because I'm superstitious.
I want to be sympathetic towards her, so I've gained some weight and a little excess bloating. Yeah. Where's your shoe?
Video hooked up to this microscope yet?
Yeah.
In the Denver University Hospital, Lisa's eggs are collected from her ovaries.
Shake up the second half. Dr. Hurst said she had 21 follicles, and generally he does fairly well. We generally get 1 egg per follicle. There's another egg.
Happy with it?
Lovely. Oh, they're just pretty. Now, I tell you, this took some doing. Come here. Sticky cells, sticky cells, sticky cells. Come here. Let go. Let go, let go, let go, let go, let go. Right. Right. Our fertilization rates are much, much higher if we leave some cumulus. And looks like he's got great sperm, so I'm not too worried about leaving a little extra cumulus on her eggs.
I want to know how many eggs I got.
Count so far, Barb?
14.
14. So 14 so far, and they'll be counting for a little bit. So we'll roll you down to the recovery room, and I'll let your husband know that you did great. How many?
15. We'll keep yelling them out to you.
Yeah, that would be good. 15, Lisa. Still counting. Nicole?
Nauseous.
Did you hear
There's
everything
21
like that?
eggs so far.
Yeah.
They're still counting. I got 21 eggs for you. Thank you. Hopefully more. You dropped it? Did you tell Mommy to
With approximately 48,000 sperm per egg, we use a 10 microliter volume. It's fairly small, but there are a lot of sperm in there.
Sometimes a single sperm is placed directly into each egg, but in Denver, they favor a more natural method. Some 48,000 of Jack Nash's sperm are sprayed over each of Lisa's eggs, 22 in total. After fertilization, the embryos need 3 days to divide.
You're a very good mommy to Monica.
Will any of these be a marrow match for Molly?
Is she night-night? Yeah. It's been good. Everything's been going in the right direction. It's just kind of hard waiting. Sometimes it's like, I don't know if I can take anymore. The not knowing. You don't have any control over anything. You have no control. And then this morning, I guess it's just hormones. You go through menopause 25 years too early, and then they put you into overdrive. This morning I woke up and I was just crying. Everything made me sad. But it never hurts to have a good cry, right, Molly? And you know, God's gonna do what he wants to do. He'll give us a baby if he wants to, so.
2s and 1s are what you want to see. Let's see, this one is a 4-cell grade 3.
Snowing everywhere, right?
So when is he gonna call?
There's number 10, and nothing. Didn't mature. I don't think she's gonna be too happy with this.
I'm gonna put my blood pressure through the roof. Hold on. Hold on. Okay. Okay, I can. How do we do this? Hello?
Lisa? Hi, Lisa. This is Kathy Tucker calling from the IVF lab.
Uh-huh.
How are you feeling today?
Until about 10 minutes ago, I felt okay. Now I want to throw up.
Can you do this?
And some more could die by tomorrow, too. Okay, so now what? Thanks. Yeah. I can't ever do anything right, and this just isn't any different. Okay. Yeah. Thanks. Bye. 3 of them died.
Only 11 of Lisa's 22 eggs developed to biopsy stage. They had hoped for more. Once removed, the delicate single cells have a plane to catch to Mark Hughes in Washington. The sooner the cells reach Dr. Hughes, the sooner the diagnosis can start, and the sooner the right embryos can be identified for implantation. The longer the embryos remain out of the womb,
Hello, Dr. Hughes. This is Dr. Cotta. I'm calling you from the Denver airport. The sample has left on the 11:30 flight from Denver to Washington, Dallas. You should be able to get that sample around 5:30 at the airport. Good luck.
they made it, 11 tiny cells. This will be the first genetic selection ever made for two purposes. To screen for the marrow match, multiple places on the genome have to be scanned.
This is the most difficult diagnostics we've done. And we have to do all of this in this short period of time that we have in order to make the diagnosis in time to transfer the embryos. It's pushing the limits.
What are the chances of success from 11?
We would predict that if everything goes according to plan, 8 of them would be free of Fanconi anemia. And of those 8, 2 of them will also be an identical bone marrow match for Molly.
First stage, the thermocycler. By morning, this machine will replicate each cell's DNA millions of times.
It's rolling. Sleep tight. All right, embryo 1 is a heterozygote. That means unaffected. Normal, mutant, like mom and dad. Embryo 2 is also a heterozygote. Great! Embryo 1.
As hoped, 8 of the 11 embryos do not carry the Fanconi disease. But what luck with the marrow match?
Is a 2. Got one.
Their good luck held. There was a second marrow match among the healthy embryos.
10. Those are the 2. This is great. We've got some embryos to transfer. Now we have to...
In Denver, the IVF team stands by.
There they are. That's it. Don't drop them.
No, it wasn't Kathy.
Oh, okay.
Yeah.
No, I'm silent as the night.
You may be able to see a little starburst type image on the... Okay.
Yeah?
Where we put them, they stay.
Now,
they
what
stay
happens
there.
if they implanted really, really close together? That wouldn't hurt them?
Not at all.
Okay.
Not at all.
This isn't quite how you usually do it, but...
No, that's okay.
Oh my God. Oh, please be healthy, guys. Please be healthy. Please be strong. There she is! There's Mama. What's your tummy, Mama?
Oh, the tummy's
There's Mama. Do you know what I have in my tummy? I have a baby brother or sister.
Thank you.
I have 2 little babies in my tummy.
The first embryos ever selected, both for health and for trait, are out of the lab and into the womb. Has the door to designer babies been opened just a bit wider?
It is likely that the field of genetics will proceed to a point where things like height, color of eyes, longevity, the sensitivity to certain diseases, all those things which are probably genetically determined can probably be assessed in one cell.
Are we ready for the ethical questions that genetic selection for individual traits is about to thrust upon us?
Once you get to the situation where it is essentially a complicated process about whether your offspring may or may not inherit a certain characteristic, that is aggression or gayness or some aspect which may be less preferred but not at risk for survival. You are taking decisions which are generally beyond our capability of making simply.
In selecting for traits, are we taking the first step to designer children? So far, genetic diagnosis has prevented hereditary diseases in a few hundred children. Very soon, this technique will affect millions of us. It's estimated that every single one of us has some genetic flaws. Mate with someone who carries a matching fatal flaw, and any of us could have a dying child. Already, genetic scientists can tell some people what diseases lie ahead of them, even what they are likely to die of. This young man knows he will probably die in 15 years or so of Huntington's disease.
My uncle had died from that. My grandmother had died from that. In my family, my mother showed when I was 42.
He and his wife asked Dr. Mark Hughes to help them have babies free from the Huntington's curse.
I'm pregnant with twins, a little boy and a little girl. I'll be 24 weeks tomorrow, so about 6 months pregnant.
This couple watches their healthy babies develop, knowing the father will not see them grow to adulthood. This man is unusual. He knows his genetic future. Soon, everyone could.
There's certain people you can tell, there's certain people that you can't tell. My doctor, normal doctor, doesn't know.
He cannot tell his employers.
I'm going to continue in my job, until I start showing. If they had the information that I am a carrier, to play it safe, they might remove me from that position.
He dare not tell his health insurance company.
An insurance company may not want to pick you up or something, knowing they're going to have a big bill ahead. So it's more than Huntington's. I mean, there's a lot of other things out there.
When we can predict what babies will die prematurely, even before they are born, will it be right to weed these embryos out?
I think it will be better if you can make a baby which will never have cancer, never have a heart disease. It's a baby which is going to become an adult and will live a very long time. If you don't get in if cars are also being redesigned, will not have a car accident. Longevity issues and genetics and reproduction are related in a certain way, and those things will get together to make individuals, to produce individuals which will, I think, be a lot better off than we are.
Genetic diagnosis is arguably the biggest scientific breakthrough of the 20th century. But genetic diagnosis is also a kind of time travel, and time travel has never been possible before.
We've never been able to look into the future in any aspect of our lives that I've ever known about before. This knowledge will allow us to be able to essentially predict the probability of the future. And I think that nobody has really synthesized what that means.
If our forefathers could have traveled in time and selected out embryos they knew would suffer from dreaded diseases or other inherited defects, who are some people that might not have been? No John F. Kennedy, no Steve McQueen or Albert Einstein, John Huston, Theodore Roosevelt, Ludwig van Beethoven, Charles Dickens, Charles Darwin, no Woody Guthrie, no Abraham Lincoln, no George Bernard Shaw. Can any of us be certain that we would have made the genetic cut? Then again, can we afford to lose the promise of genetic medicine? The door to genetics has already been opened. It will undoubtedly play a role in our future. We must always be careful with this powerful tool and prevent our genetic dreams from becoming nightmares.